Hey guys, welcome back to the channel. Hope you guys are doing good. This is part 3 of the video on cell injury and its pathology. So without wasting any more time, let's get started. In previous part of the video, we discussed about necrosis and its types. In this video, we will discuss about apoptosis. So the first question which comes in our mind is, what is apoptosis? Apoptosis is a Greek word which means falling off. It is physiological as well as pathological cell death and is not associated with inflammation, unlike necrosis, which is associated with inflammation. The hero of this process is the mitochondria because it plays an important role in the process of apoptosis, which we will discuss later in the video. Apoptosis occurs as an active process, which means there is usage of ATP. It is a programmed cell death, also known as caspase-dependent programmed cell death. You may wonder where this caspase came from and what is it. So, to understand it better, let's make a mnemonic for it. We can write caspase like this. The end has A's in it, so it is clear that it is an enzyme. This C denotes the cysteine residues which are there in this enzyme. And this ASP can be understood as whenever this enzyme sees aspartic acid residues in a molecule, it breaks the molecule after those residues. Now, let's see some examples. As we discussed earlier that apoptosis can be physiological or pathological. And we know that physiological apoptosis is normal. The example of physiological apoptosis include organogenesis, that is during fingers and hand formation. So what happens is, during embryonic time or the time of formation of fingers and hands, the physiological apoptosis takes place, which creates the normal web-like pattern of our hands. If this physiological apoptosis doesn't take place during the time of organogenesis, it will lead to a condition known as syndactyly, where the fingers are joined. Another example of physiological apoptosis is endometrial shedding, which occurs during the time of menstruation. The examples of pathological apoptosis can be seen in cases of viral hepatitis. Most commonly, hepatitis C, where the characteristic features are the Councilman bodies. Or in the case of a dermatological problem known as lichen planus. Or in the case of a dermatological problem known as lichen planus, where cytoid bodies are seen. And to be clear, pathological apoptosis is not normal. Now, let's see how this process is carried out and what is the mechanism of apoptosis. So, there are two processes involved, known as initiation and execution. Initiation is caspase 8, 9 and 10 dependent. And execution is caspase 3, 6 and 7 dependent. Initiation is further classified into two pathways, extrinsic and intrinsic pathway. Let's discuss each of these separately. Starting with the extrinsic pathway, also known as death receptor pathway. This process starts by an apoptotic cell or the cell which wants to die. These cells express receptors known as CD95 or FAS. Corresponding to this, the T cell has CD95 ligand or the FAS ligand. And you can make it out yourself that this T cell binds to this CD95 or FAS on the apoptotic cell. This leads to trimerization of this CD95 or FAS on the apoptotic cell. After this process, there is something known as FAS-associated death domain, which activates the procaspase 8 and 10 into caspase 8 and 10, which are required for apoptosis of the cell. This process of activation of procaspase into caspase is inhibited by a molecule called FLIP, which is an anti-apoptotic molecule. These are the same steps in the theoretical form. You can pause the video and read them. You may wonder where is caspase 9, because we discussed here that initiation is caspase 8, 9 and 10 dependent. So, to understand the activation of caspase 9, we need to understand the intrinsic pathway, also known as the mitochondrial pathway. When a cell is exposed to some kind of stress, like UV rays, there are certain stress sensors present in the cell, like BIM, BID, BAD, NOXA and PUMA, which get activated and increase pro-apoptotic factors and decrease anti-apoptotic factors. The pro-apoptotic factors include P54, BAC, BAX, and BCL excess. Whereas the anti-apoptotic factors include BCL2, MCL1, and BCL-XL. Now what happens is these pro-apoptotic factors make the mitochondria release two things, which include cytochrome C and SMAC and Diablo. The cytochrome C combines with the apoptosis activating factor 1 or APAF1, and this combination of cytochrome C and APAF1 is called as an apoptosome. This apoptosome is responsible for activating the procaspase 9 into caspase 9. With this, we solved our query regarding caspase 9 activation. We completed the process of initiation and move on to the next process, which is execution. So after the initiation process, the caspase 8, 9 and 10 activates caspase 3, 6 and 7. These caspases in turn activates three enzymes in the cell known as phospholipase, protease, and endonuclease. As you already know from our previous videos that these enzymes break the three most important parts of the cell: the membranes, proteins, and the nucleus. These enzymes break the cell and the resultant small bodies are called as apoptotic bodies, which are membrane-bound bodies containing the cell fragments. These apoptotic bodies give a signal to the macrophage, and the macrophages engulf these apoptotic bodies and carry out the process of phagocytosis. You may wonder what kind of signal do these apoptotic bodies send. These signals consist of something called as CQ1, thrombospondin, and phosphatidylserine. As you can see, I have highlighted the phosphatidylserine in red because it plays the major role in these signals. In a normal cell, the phosphatidylserine is present on the inner layer of the membrane. You know from physiology that in a normal cell, the cell membrane is a phospholipid bilayer. And in that case, the phosphatidylserine is present on the inner layer of the membrane. In the case of apoptosis, the phosphatidylserine is flipped to the outer membrane. And this gives the signal to the macrophages to start phagocytosis. With this, we complete both the pathways and the process of apoptosis. So that is it for this video guys. Don't forget to subscribe the channel and follow us on Instagram. Links in the description.